Thank you, Sarah, for that very kind introduction and for inviting me to speak here today. So I will spend most of today sharing some our recent work on characterizing the in vivo latent HIV reservoir. That was the paper that you guys had discussed. However, we, like many other HIV virologists, pivoted to doing SARS-CoV-2 work when the pandemic had hit. And since most of the new data that we had generated over the past year or so was limited to SARS-CoV-2 work, I thought that in the end, I would also share some of our recent COVID findings. And so let me share my screen now. Okay, I'm going to assume that you guys can see my screen. I'm going to do it in presenter mode. Speak up if you don't see it, but I'm just going to assume that everything is going as it should be. Okay, so okay, thanks. Thanks for confirming. Okay, so to begin with, I thought I would start by saying a bit about what in general my lab focuses on. And so the folks in my lab, we are all interested in this fundamental question of what makes some cells more susceptible than others to HIV infection. Now, we all know that CD4 positive T cells are one of the primary targets of HIV, but not all CD4 positive T cells are equally susceptible. And so we are interested in understanding both the cell intrinsic factors as well as cell extrinsic or external factors that will determine whether a particular CD4 positive T cell becomes infected or not upon exposure to the virus. And the lab has been investigating both of these aspects. With regards to extrinsic factors, we've done quite a bit of work looking at how the mucosal environment can in fact greatly promote HIV infection of target cells. And with regards to the intrinsic factors, that's where we've been taking advantage of a lot of single cell analysis tools like Cytometry, which I'll say more about in just a minute, to characterize what is it about the intrinsic state of those CD4 positive T cells that are most or least susceptible to HIV infection. And when we look at infection, we'll be looking at both productive infection as well as latent infection. Okay, and so this will be the topic I will be focusing on today. And so when we began the project several years ago, we were interested in this basic question of amongst a diverse population of CD4 positive T cells represented here by these different colors, which ones are the ones that are most susceptible to HIV infection and why? And the source of these CD4 positive T cells can be from blood or they can be from tissues. And we started off with tonsillar CD4 positive T cells because tissue cells are obviously important in the context of HIV pathogenesis, but also there's a subset of tonsillar CD4 positive T cells that are susceptible to HIV infection in the absence of ex vivo stimulation. And so we wanted to ask, okay, so amongst this diverse population of CD4 T cells, which ones are the ones that are susceptible to HIV? And in this little cartoon here, it would be that these blue and yellow cells are the susceptible ones, whereas the others are not susceptible. And so the technology that we decided to use to really investigate this in depth was Cytometry. And so I think many of you are familiar with Cytometry, but for anyone that's not, essentially it is just a single cell protein quantitation tool that is analogous to flow cytometry, but it uses a mass spectrometer to read out the data instead of a flow cytometer. Now, one advantage of Cytometry over conventional flow cytometry is that because you don't have this issue of spectral overlap, many more parameters can be simultaneously monitored. And what this essentially allows for is a much higher resolution of the immune system. And it also allows us to apply various bioinformatics tools to analyze the resulting data sets. And so shown here is an example of one of the Cd4 T cell panels that we had developed to characterize susceptibility of cells to HIV infection. And so typically we have a way to unambiguously identify the Cd4 T cells. And then once we've identified them, we can distinguish them based on various differentiation and activation markers. We also usually include a lot of cell adhesion and homing receptors, particularly when we're working with tissue cells. And we also have a lot of intracellular factors as well that can help us distinguish the various polarization and differentiation states of the CD4 T cells. And so So when we started, we did an ex vivo infection system. And so we obtained tonsillar Cd4 T cells from uninfected individuals, and we exposed them to a transmitted founder reporter virus that we had. And we wanted to ask, what are the cells that are most susceptible to HIV infection? And these are results from one of the early experiments that we had done. I'm showing the results in the form of a t-SNE. For any of you that aren't familiar with looking at t-SNE, essentially it's just a two-dimensional depiction of high-dimensional data. And in this case, the high-dimensional data are 4D dimensional CyTOF data sets. And so in a t-SNE, each dot generally corresponds to one cell, and two cells that are closer together in high dimensional space, they appear closer together on this two-dimensional plot. And so these cells end up forming these islands, if you will. And so the islands are essentially, cells that are more similar, phenotypically similar to each other. And so these here are the uninfected T cells in a particular donor specimen. And then what we did was we also looked at the HIV infected cells, and we could identify these cells because we had a surface marker to identify those cells that were productively infected. And when we looked at the productively infected cells, this is what we observed within the same t-SNE space. So what you can see is that most of the infected cells fall within this empty region here that's not occupied by cells in the uninfected sample. And so what this suggests is that the infected cells are phenotypically distinct from anything in the original culture. And we give a name to this phenomenon. We call it viral-induced remodeling. And these cells are considered quote unquote remodeled because they're phenotypically distinct from anything in the original culture. Now, the fact that HIV remodels cells suggests that this little cartoon I had showed earlier, it's not that accurate, because it's not the case that these infected cells are simply a subset of the starting population, but rather they changed a bit. And so I tried to depict this change by changing the colors here from blue and yellow to purple and pink. And just to put this in the context of the t-SNEs I had showed earlier, essentially the uninfected cells look like this, and the productively infected cells look like this. But we are really interested in what are the phenotypes of these cells. These are the ones that are preferentially targeted for HIV infection, and they are the cells that you would essentially want to protect from infection. So how do we kind of get this information here? And so what we decided to do was to take advantage of the high dimensional nature of these CyTOF data sets to essentially predict the original state of these cells before HIV-induced remodeling. And this was an approach that we gave the name of predicted precursor as determined by SLIDE, or PP-SLIDE for short. We first described it in this 2017 paper. And I won't go over the details of the approach, but essentially the concept behind it is really pretty simple. What we do is we consider the uninfected cells the control cells essentially as the atlas of all the different types of uninfected CD4 positive T cells that are present in a particular sample. We then take the high dimensional information amongst our infected cells and match it against this atlas to identify the phenotypically most similar cells. And those cells serve as the predictive phenotypes of the original cells targeted for infection before HIV-induced remodeling. And this approach generally works because although HIV infection will change the phenotype of the cell, it typically doesn't change it so much so that that cell has completely lost its original identity. And furthermore, because immune cells come in subsets, there is typically a cell of the same subset in the uninfected sample. For example, if there was an infected Th1 cell here, there is going to be somewhere in the uninfected culture an uninfected Th1 cell. And so these cells here, we gave it a name. We called it the predicted precursor cells. Predicted because we are predicting their phenotypes, and precursors because they are the precursors to these productively infected cells. And so in our first publication using this kind of methodology, we identified various subsets known to be preferentially susceptible to HIV infection, including T follicular helper cells, Th17 cells, as well as others that weren't previously described. But we could validate these predictions by sorting out these subsets ahead of time and then exposing them to our reporter virus and showing that they were indeed more or less susceptible to infection as we had predicted. Now, more recently, Tong-Sui Ma, who is a very talented postdoctoral fellow in my lab, she wanted to use this approach to ask the question of what about the initial site of HIV transmission in women? So women comprise more than half of those that are newly infected worldwide, and yet very little is known about the initial cells targeted for HIV infection in the female reproductive tract. And so we decided to essentially use this approach to better understand that. And so Tong Cui started off by doing a flow cytometry assay. And this was what made us interested in examining genital T cells. And so what she did was she took PBMCs, either unstimulated or she stimulated them with a mitogen to get them to be more permissive to HIV, or she took tonsil cells, which we abbreviate as HLACs, or genital T cells. So these are genital T cells from HIV uninfected individuals that have been processed into a single cell suspension. And then she exposed them to our reporter virus, which is a transmitted founder reporter virus. And so the infected cells are shown within the gate here. Infected cells are HSA high because that's our reporter, but they are also CD4 low, and that's because productive infection by HIV is known to downregulate cell surface CD4. And so what we found was that when we expose unstimulated PBMCs with our reporter virus, we have a low proportion of infected cells, consistent with unstimulated PBMCs being, for the most part, somewhat resistant to productive infection. But if we pre-stimulate the PBMCs ahead of time, you can see we have almost a 10-fold increase in infection rates. Now, when we use tonsils instead of PBMCs, we do see almost 5-fold increase in infection rates in the tonsils relative to the unstimulated PBMCs. That's consistent with what others have reported, that these tonsillar cells are more susceptible in the absence of stimulation. But what was very striking was that when we looked at these genital T cells, we saw they were way, way, way more susceptible to infection. You can see in this particular culture, more than 25% of the Cd4 T cells in the culture became infected when we exposed them to the same viral inoculum as these other cultures. And when we looked at a bunch of donors all together, when we looked at the data all analyzed together, we did indeed find that these endometrial T cells were much more susceptible to HIV infection. You can see more susceptible than both types of PBMCs as well as the tonsils. There's actually a break here, so you can see there's actually a lot more infection in these endometrial T cells. And so this made us really interested in investigating, what is it about the features of the endometrial T cells that are very susceptible, and also does HIV remodel these cells upon infection, and if so, how might that affect subsequent spread of the virus? And so I want to spend the majority of today's time talking about the in vivo reservoir cells. So I won't go over the details here. If you're interested, you can read more about it in this paper that we published last year. But I will sort of give the take-home message, which is the model that we came up with. So what the data suggested after we did our CyTOF and PP slide analyses together was that these endometrial T cells are highly, highly susceptible to HIV. And that upon HIV infection, HIV actually remodels these cells extensively. They remodel it by downregulating components of the T cell receptor complex, which we think can dampen the ability of a proper adaptive immune response against the virus. And we also saw upregulation of BRCA5, which is a factor that's been shown to promote survival of HIV infected cells. And we think this serves the purpose of promoting the survival of the HIV infected cells long enough so that they can migrate to other sites that have an even richer source of CD4 positive T cells. And indeed, we saw a lot of remodeling that was consistent with the ability of these cells to leave the genital tract and migrate into the lymph nodes. For example, upregulation of the lymph node homing marker CCR7, and also upregulation of CXCR5, which is very interesting because, as many of you know, CXCR5 drives cells into the follicles, and the follicles of lymph nodes are known to have a lot of HIV susceptible cells. And so we think that this is how an initial beachhead of infection can really result in systemic infection quite quickly. Okay, so that was one use of a PP slide and CyTOF analyses to essentially look at susceptibility of cells in an ex vivo infection system. And that particular study, we had used we had looked at just the proteome of HIV susceptible cells and how the proteome was remodeled by HIV. But more recently, we, together with our collaborator, Mohammed Abdelmohsen at Wistar, we've also been interested in looking at the glycome of HIV-infected cells. So in this collaborative study that we published last year, led by their group, we found that cells harboring active HIV transcription, that they had a different glycome, a cell surface glycome. In particular, we found increases in fucosylation, as well as some other sugars. And this study, however, and glycomics in general, has been done at the bulk level, but we thought that it would be really valuable to essentially look at it at the single cell level, but the tools didn't exist for that. However, we reasoned that just as we can use antibodies in CyTOF to detect specific proteins, that in an analogous fashion, we can use lectins, which are specific proteins that bind specific glycan or sugar patterns. And so essentially what we decided to do was work out conditions where we conjugated the lectins to metal lanthanides and then mix them together with various antibodies. And so this allows us now to have a CyTOF panel that allows us to look at not only different proteins, but also different types of sugars. And so in the lab, we casually call this sweet CyTOF because it's recognizing sugars. And essentially, we've recently conducted a big study where we've compared HIV infection of resting PBMCs, activated PBMCs, tonsils, as well as genital T cells, looking not just at the proteome, but also at the glycome using the sweet CyTOF panel. And I won't go over this too much, but I'll just say that, just as an example, one thing that we found that memory CD4 positive T cells expressing high levels of the glycan sialic acid tended to be more susceptible to HIV infection compared to those expressing low levels of sialic acid. And so that came out of the CyTOF and PP slide prediction, but then we were able to validate those predictions by sorting out these populations ahead of time and then showing that these populations were indeed more or less susceptible to HIV infection as the original CyTOF and PP slide had predicted. And so this is a study that we're trying to wrap up right now, and hopefully we can get it into the public domain soon. But now what I want to do is talk about how we've also been using this kind of PP slide approach to not only look at, an ex vivo infection system, but also to look at the in vivo HIV latent reservoir. And this is a collaborative project that has involved many folks in my lab, as well as many collaborators outside of the lab. And the fundamental question we wanted to address was, what are the phenotypes of in vivo HIV reservoir cells? Now, we all know that one of the main barriers to an HIV cure is the small but persistent population of latently infected cells. And there are a number of reasons why it's been so difficult to eliminate or control this latent reservoir. One being that as a field, we still don't have a good understanding of what types of cells in vivo harbor latent HIV. And one of the reasons for this is that there is no universal cell surface biomarker that can distinguish these cells from a regular uninfected CD4 positive T cell. And so what this means is that when you have a patient sample and you want to know what cells there are latently infected, you have to take that entire sample, stimulate it with a mitogen such as anti-CD3, CD28. And what the stimulation will do is it will cause some of the latent cells to start producing viral proteins, which you can identify at the single cell level. And so you can characterize this cell extensively, for example, by FACS or CyTOF. And this cell here is now referred to as the reactivated cell. It was once latent. But the important thing to note is that when you're characterizing this reactivated cell, it's not the same as the original latent cell. And there are a number of sources of that change, one being that we've stimulated the cells ex vivo. So you'll have, for example, upregulation of a lot of activation markers. And the second reason is that, as I mentioned earlier, HIV replication itself in the cell will induce significant remodeling of the cell. And so how do we overcome this issue of the cells which we can identify and characterize, these reactivated cells being different from the cells that we actually want to characterize, these original latent cells? And so we, of course, apply the principles behind PP slides in our ex vivo system to now try to predict the original state of the latent cells before ex vivo reactivation. And so in the lab, we like to refer to this as the molecular facial recognition project. And I think it could be helpful to think of this project in the form of an analogy, and specifically that of aging. And so some of you may have kind of seen me bring this analogy up before, but essentially the analogy here is that this photograph here on the left-hand side is a picture of a somewhat younger Sean Connery. And then the one on the right-hand side is a photograph of a somewhat older one. The analogy here is that the younger one corresponds to our original latent cell, and the elder one corresponds to our reactivated cell. And so, as I mentioned, we have the information here on the right-hand side. We can identify these reactivated cells, but the information that we want is that on the left-hand side. And so how do we get that? So essentially, with the PP slide approach, what we do is we first establish a patient-specific atlas of all the different types of unstimulated CD4 positive T cells present within that participant sample. And then we take the high dimensional CyTOF information here and match it against this atlas to identify the phenotypically most similar cell, which is this one here. And this serves as the predictive phenotype of the original cell targeted for latent infection. And so just to summarize the approach, essentially we identify our reactivated cells and then we match it against this atlas using what's called a k-nearest neighbor approach. And then we identify the phenotypically most similar one. And then as a final step, we compare the features of that predicted latent cell to other cells to try to identify unique features associated with latency. Okay, so to start, we wanted to first do a proof of concept experiment to demonstrate that this approach can indeed work to identify precursors of reactivated cells. And so we started with a simplified system of JLATs. And so JLAT is a latently infected cell line that originates from GRCAS. And because different clones of JLATs exist, and because these clones were generated and propagated by different labs, distinct phenotypic properties developed in these different clones over time. And so we wanted to ask the basic question of, given two different clones, one called 6.3 and one called 5A8, if we stimulate one of them to reactivate latent HIV, for example, the 6.3, can we, given the information here, predict that these cells came from these ones when given a choice of these two? And so we went ahead and phenotyped all these populations by CyTOF and then analyzed the data by ppslide, which I'll show right here. And so I'm depicting the data in the form of t-SNEs. And so first here, I showed the unstimulated 6.3, which reside mainly in the lower left-hand region here, and then the unstimulated 5A8s, which reside in the upper left-hand region here. And the fact that they are in distinct regions of the t-SNE suggests that they are phenotypically distinct, even though they came from the same parental line. Now, the stimulated 6.3 cells are down here. You can see it's in a third unique region of the t-SNE, suggesting that reactivation does indeed change the phenotype of the cells. So now the question is, given the information here, can we predict that these cells came from these ones when given a choice of these two? And so we took every cell here and matched it against a combined or concatenated atlas of all of these events, split at a 1-to-1 ratio. And we asked how often it picked from here as opposed to here. And shown here are the resulting nearest neighbor cells. And you can see that it looks quite similar to this one and not so similar to that one. And in fact, there was 99.2% overlap with the correct one and only 0.8% overlap with the incorrect one. And so we consider this an error rate of 0.8%, which we were quite happy with, given that these are clonal lines that originated from the same parental T cell line. Okay, so having done that proof of concept experiment, we then wanted to move on to participant samples. And for this, we worked with the SCOPE cohort at UCSF. And so this is one of the first participant samples which we analyzed. And so we got leukapheresis specimens from this participant and immediately established the atlas of unstimulated CD4 positive T cells with a subset of the sample. And so this is actually an atlas of the unstimulated memory CD4 positive T cells. We had pre-purified the memory cells ahead of time to further enrich for the inducible reservoir. And then with the remaining cells, we stimulated for 40 hours of hematoamycin, and we identified 20 reactivated cells. And we identified these reactivated cells by using multiple antibodies against GAG. And so this is shown here, where if you don't stimulate, you don't have anything in the double positive, but when you stimulate, you have a clear population here of this kind of dual GAG positive population. So we identified 20 samples. We identified 20 sorry, is there some feedback? Okay, I don't hear anymore. Okay, so we identified 20 cells, 20 reactivated cells. And when we put them onto this t-SNE plot, this is where they fell. So this is actually 20 cells all stacked on top of one another. And the fact that they are in a unique region of the t-SNE suggests that indeed reactivation does change the phenotype of the cells. So now to assess where these cells would fall within the t-SNE, the atlas of the t-SNE, we conducted our ppslide analyses, and this is where those cells ended up. And this here is an overlay of the data. And so shown on this next slide are some of the ways that we've been looking at the data. So we have what we call a map of where these predicted latent cells are in red relative to the atlas. And what you'll notice is that it's not randomly distributed. There are clearly regions that lack these predicted latent cells, such as up here and down here. Now we can look at any particular antigen in more detail. And so here I show the expression levels of PD-1 because that's an antigen that's been shown by others to be high on in vivo latent cells. And indeed you can see based on this heat map depiction that regions that have high levels of PD-1 do include regions where our predicted latent cells are. And we've also used various unbiased clustering algorithms such as FlowSum to ask questions such as what are the clusters that are most enriched for these latent cells, and what are the phenotypic features defining these particular clusters. Now, one main benefit of our data sets is that we have 40 parameter information on our predicted latent cells. And because many of these parameters are cell surface proteins, we can, based on this information, design a sequential sort strategy that we think in the end should enrich for latent cells. And so we did that here for the same donor that I described earlier, where I'm showing the latent cells in red and then the atlas cells in gray. You can see our latent cells tend to have low levels of both CD38 and CD57. They tend to have high levels of PD1. I already showed that in the previous slide, but interestingly, the other T follicular helper marker CXCR5 tends to be low on these cells. And here are just some other markers that tend to be higher or lower on these predicted latent cells. And so with the sequential sort strategy, we can now end up with a population that we predict should enrich for these latent cells. Now, as a comparison population, this particular donor, we selected this population here, which we call a disenriched population. You can see there's only one red cell here, so it should be disenriched for the reservoir. But an important thing I want to point out is that this disenriched population is downstream of the PD1 high sort strategy. And we did this on purpose because PD1 is one of the most well-validated markers of the in vivo reservoir. And we wanted to prove to ourselves that we could essentially bifurcate this, that we could do even better than just selecting for PD1. So we pre-selected for these PD1 high cells. And so with these two sorted populations, we then did a viral outgrowth assay to ask whether more virus grew out of the enriched one. And so this is a modified QVOA. We weren't able to do a bona fide QVOA because when you sort based on that many parameters, you just don't end up with enough cells in the end. But essentially, this is a modified QVOA. In this particular viral outgrowth assay, what we do is we stimulate the cells in bulk for 2 days with anti-CD3, CD28. We then dilute the cells down to 40,000 per well. And then we add in these molt CCR5 susceptible cells, and we propagate the cultures for about 2 weeks. And at the end of 2 weeks, we do a P24 ELISA to ask whether more virus had grew out of the enriched population compared to the disenriched one. And the answer was yes. And in fact, we had almost 3 orders of magnitude more virus grow out of the enriched one compared to the disenriched one. This is a long scale here. And so we were quite happy with these results, given that even the disenriched one was predicted to be pretty enriched to begin with, given that they were PD1 high cells. And so this is one of the first donors that we conducted such a validation assay on. This is just another donor that we went ahead and did a similar kind of validation where we designed this particular sort strategy to end up with this population of cells predicted to be enriched for the reservoir. And then here, because we had more cells, we could compare to 3 different disenriched populations. So these were the 3 different populations predicted to be disenriched. And what I want to point out is that all these cells here, which I've boxed in pink here, they are downstream of the PD1 high sort strategy. And again, we picked a number of disenriched populations that were PD1 high for the reasons that I had mentioned earlier. And so we did a similar kind of viral outgrowth assay here, except this time instead of just diluting down to 40,000 per well, we diluted to different numbers of cells per well. And so first I'll draw your attention to the enriched population here, where we added either 1,600, 8,000, 40,000, or 200,000 cells per well. And you can see as we added more cells into each well, we had a higher likelihood of outgrowth. And this is reported both in terms of the concentration of P24 in each of the 4 wells for each condition, as well as the number of cells that were positive out of 4. And so, when we added only 1,600 cells, nothing grew out. But as we added more and more, you can see there's more and more virus that had grown out. But importantly, this was only observed in the enriched population for all 3 of the disenriched populations, including the 2 that were pre-selected on PD1 high cells, that even at the highest concentration of 200,000 cells per well, we didn't see any viral outgrowth. And also, if we just take the starting population of memory CD4 positive T cells, we also didn't see outgrowth. So this suggests that markers identified by our Cyte Health PP slide approach can enrich for the replication competent reservoir. We've done this kind of validation assay on 4 different donors, and I'm showing the cumulative data here, where in all cases I'm showing the enriched population in this sort of peach color, and then the disenriched population, both of which are PD-1 high in the purple. And you can see that the enriched population consistently grew out much more than the disenriched one. And most of the time, this enriched didn't grow out at all, and this one it grew out a little bit, but you can see it's still less than the enriched one. And so this was kind of nice validation that these surface markers really could enrich for the replication competent reservoir cells. And so this was useful because we now had a way to enrich for these replication competent reservoir cells. And so we can ask various mechanistic questions about, what are the features of this, pre-sorted population of cells that's much enriched for replication competent cells. And so some of the stuff we've done is, for example, collaborate with Steve Ukel's lab to look at, these enriched populations, are they transcriptionally active for HIV transcripts? And so these are the same populations I showed on the previous slide. And then overall, we find that the enriched populations, which have more replication competent HIV, that they tend to have more transcriptionally active HIV, suggesting that the transcriptionally active reservoir may be an important one to target. We've also collaborated with Sarah Palmer's lab at University of Sydney to do proviral sequencing on these specimens. And this is just an example of one of the donors we've analyzed, where we're comparing an enriched population here to one of the PD-1 high disenriched populations. And you can see in the enriched one, in this particular donor, we found that 65.2% of all the proviruses that we pulled out were fully intact, which I'm told is a very high percentage. And this amounts to 214 fully intact per million Cd4 T cells, whereas for the disenriched population from the same donor, that we only found 0.9% of the proviral sequences were fully intact, and this corresponds to only 2 per million. And so we can also look much more deeply into these results. For example, we've looked at these particular sequences, and we find that these fully intact sequences from this enriched population is interestingly made up of 2 main groups of clonal expansion shown here. And so we're digging much more deeply into the data now, and also with this particular participant, we're actually doing a longitudinal analysis to see how these kinds of things change over time. So some other aspects that we can take advantage of now that we have the ability to do this CyTOF slide approach to chart the in vivo latent reservoirs, we can ask questions such as whether latency reversal agents, LRAs, reactivate different subtypes of reservoir cells as compared to broad spectrum mitogens such as PMA anamycin. And this is an example of one experiment where we investigated that. And so what we did was we had one participant come in 3 times, and at the first time point, we stimulated his cells with just PMA anamycin. At the second time point, we stimulated with either PMA anamycin or the LRA combination romidepsin together with ingenol PEP005. Then at the third time point, we stimulated with just the LRA combination. And then here I've put all the data onto the same t-SNE space. And what you'll kind of visually notice is that the PMA stimulations result in reactivation of more types of reservoir cells. In particular, you can have this region here in the upper right-hand side that seems to be missing with the LRA combination. Indeed, using statistical approaches, we showed that the 2 PMA anamycin samples from different time points are actually more similar to each other than the PMA anamycin versus the LRA combination within the same time point, suggesting that different combination of LRAs may be necessary to really capture reactivation of the entire full spectrum of the subsets of reservoir cells. And so everything I've shown thus far is all blood, but we have looked at tissues. And so this is an example of one of the tissue experiments that we did. And we had one participant come in again for 3 different study visits, where at the first visit we obtained just blood, and then at the second 2 visits we obtained lymph node aspirates, so fine needle aspirates or FNAs, as a source of lymph node cells. And then when we analyze all of these data together, interestingly, we find that the lymph node reservoir cells from the 2 different time points actually resided in somewhat similar areas of the t-SNE. You can see the red here and here. This is actually quite a few cells. It's 28 cells here and 21 cells here. It just doesn't look like a lot because they're all kind of stacked on top of one another. But the fact that they're in similar regions suggests that over time, perhaps the tissue reservoir phenotypes don't really change so much. And we've also looked at this more extensively by looking at specific markers. And so here, this is the same participant where we have blood and the 2 FNA samples. And so we can look at various marker combinations. So here we've looked at the central memory marker CD27 and the activation/T resident memory marker CD69. And here we're dividing things into 4 quadrants. And you can see with the blood, you kind of have the red cells all over the place. But then with the lymph node cells, they tend to be mostly, or actually, if we draw the gates this way, all CD27 positive and CD69 positive. So we definitely see differences between the lymph node and the blood. And here are some other markers, CXCR5 and PD1, which identifies T follicular helper cells. Perhaps not surprisingly, the ones in lymph nodes are much more overrepresented amongst these T follicular helper cells. And here are just some other markers here as well, ICOS and TIGIT, where interestingly for the lymph node cells, they tend to be ICOS high, but TIGIT low. Okay, so we've also looked at gut reservoir cells, and this particular analysis is a cross-sectional analysis comparing gut reservoir cells from 4 individuals to blood reservoir cells from 4 different individuals. And just one thing that kind of emerged here is that most of the gut reservoir cells tended to be in quadrant 3 when we looked at CD27 versus CD69. Suggesting that they are not central memory-like, but they are T-resident memory-like, and in contrast to the blood ones where it's kind of distributed over all 4 quadrants. And in data which I won't have time to show today, even though we see a lot of differences in phenotype features between blood and the various tissues, we actually also do see commonalities between different tissue reservoirs, in particular between the gut and the lymph node. And I'm happy to say more about that afterwards for anyone that might be interested. And so one thing that we noticed in our data sets when we ran more and more individuals through our Cytometry slide pipeline is that there are shared features between different individuals. So here I'm showing the reservoir cells in red from 4 different individuals. And you can see they all kind of reside in a similar region of the tissue, sort of mostly in this upper left-hand region, maybe a little bit in the lower right. And so that suggests that there are shared features. And indeed, when we look more closely at the data, we can find markers that are consistently up or down regulated on these reservoir cells compared to the total population of memory CD4 positive T cells. And so this suggested to us that we could, based on this information, design a universal sort panel that could enrich for reservoir cells in a donor independent manner. And so we looked at all our donors together and then used that information to design this universal sort panel. And what we did was we tested this universal sort panel on 4 brand new donors that we had never run CyTOF or PP slide on. And what we did was we sorted out 2 populations predicted to be enriched. Basically, they're really similar, except one was TIGIT high and one was TIGIT low. And we did this because our predicted data suggested that in some people, the reservoir cells are TIGIT high and others are TIGIT low. So we wanted to test whether we could really confirm that to be the case. And then we sorted out one disenriched population here. And again, we picked a disenriched population that was downstream of the PD1 high sort strategy so that we could also prove to ourselves the whole PD1 thing again. And you can see in all cases, basically the cells that were predicted to be enriched for the reservoir based on PP slide did enrich for the replication competent reservoir in this viral outgrowth assay. And so we consider this some nice validation that the universal sort panel can be a useful approach to enrich for the replication competent reservoir cells. And so we think this is useful because there are a lot of really nice analysis tools out there that are difficult to apply on in vivo latently infected cells because these are so rare. In particular, single cell RNA-seq is a really nice approach, but when you have only about 1 in a million replication competent reservoir cells, it's difficult to apply those kinds of approaches. But now what we've been doing is pre-sorting ahead of time to enrich for replication competent reservoir cells and then applying not only single cell RNA-seq, but also something called antibody-seq, where we can look at surface proteome, as well as T cell receptor analysis so that we can look at the extent of clonal expansion of different reservoir cells to further analyze the data. And this was what we were working on full time when the pandemic hit, and we're now starting to resume this, but hopefully we should be able to have some interesting data on that soon. Okay, so to summarize this part of the talk, I described PP slide as an approach for characterizing the proteome and now more recently the glycome of HIV susceptible cells that are infected ex vivo. And one take-home message is that HIV remodels genital cells extensively to promote their survival and dissemination. And then after that, I talked about using the same PP slide approach as one to chart the phenotypes of in vivo latent cells. And some overall conclusions from this analysis is that that the in vivo latent cells are heterogeneous, but they're not randomly distributed. Importantly, we validated this method by a variety of sorting experiments. Our data suggests that latency reversal agents reactivate different subsets of latent cells compared to broad spectrum mitogens. I also showed some data comparing tissue versus blood reservoirs, and also with our ability now to enrich for the replication competent reservoir cells in unstimulated samples, we can more closely look at various mechanisms of viral persistence. Okay, so in the last 10 minutes or so, I'm going to pivot completely into some of the recent work we've been doing using CyTOF panels, Cd4 CyTOF panels, CD8 CyTOF panels, to characterize the T cell response to SARS-CoV-2. And so early on when the pandemic hit, we were interested in T cells, not only because we in general have an interest in T cells, but also because early on it emerged that T cell lymphopenia was one of the most defining characteristics of severe COVID-19, suggesting that perhaps T cells have an important role in preventing a severe disease. This is also consistent with the observation that there's more clonal expansion of T cells in moderate cases as compared to severe COVID-19. And also from literature from the closely related SARS-CoV-1, there's evidence that SARS-CoV-1 specific T cells can be very long-lived, more than 10 years. And so perhaps they could also be long-lived in the context of SARS-CoV-2. And I want to contrast some of this with the B cell and antibody literature, where in contrast to T cell lymphopenia being characteristic of severe COVID-19, interestingly, the levels of SARS-CoV-2 antibodies actually positively associate with disease severity, and the reasons for that aren't completely understood, and it's probably quite complex. But also interestingly, there are case studies of individuals that aren't able to mount effective antibody responses, and yet they can still recover from COVID-19 without intensive care, suggesting that other cells, for example, T cells, might be able to step in when antibody responses aren't sufficient. And so we wanted to look closely at the SARS-CoV-2 specific T cell response in COVID-19 patients. And so for this, I worked with Sugi Lee at UCSF. She started what we call the CHIRP cohort of outpatient individuals. So it's a longitudinal cohort where we sampled from the acute phase of infection all the way to 6 months out. And so what we did was we decided to focus initially just on the SARS-CoV-2 specific T cells. And on this slide, I show how we identify these T cells. So first, we take some of the cells and we analyze it by CyTOF. And so the baseline sample is essentially when we just take the PBMCs and then immediately analyze it. And here I'm comparing a COVID negative individual to a COVID positive individual. I'm showing interferon gamma versus CD4. And so as you can, you might expect at baseline, you don't have much interferon gamma production. Now, when we take the samples and then we stimulate with PMA onomycin, you see a lot of interferon gamma production. This just as a positive control for our interferon gamma antibody. But then now this here on the right-hand side is what allows us to identify our antigen-specific T cells. So we either treat with co-stimulation. We either treat with co-stimulation alone or co-stimulation with CMV peptides, so that we can look at CMV-specific T cell responses just as a comparison to the SARS-CoV-2 specific T cells, or we add in the SARS-CoV-2 overlapping peptides. And you can see, these two individuals are both CMV seropositive, and we see responses there. And then we have a pretty clean system where we don't have SARS-CoV-2 specific T cells detected in this COVID negative individual, but we have it in the COVID positive individual. And this is just a bunch of convalescent individuals that we had analyzed. Now, the CHIRP cohort is all outpatient. So these are all individuals that had recovered from mild infection. And the first study that we had published on this, we essentially just reported that. We didn't compare to severe hospitalized individuals. And that was published last year. And, we kind of found different phenotypes amongst the SARS-2 specific CD4s versus CD8s, where the CD4s tended to be more central memory-like. The CD8s tended to be more temerous. But I do want to go over one piece of data from this published study, because I think it's really interesting, and that is that these SARS-CoV-2 specific T cells appear to persist for a pretty long time in these individuals. So here's one participant where we looked out to 69 days post-infection, and we can still see SARS-CoV-2 specific T cells. And interestingly, these cells at that stage still express CD127. So CD127 is the alpha chain of the IL-7 receptor, and it's involved in homeostatic proliferation. And so this was interesting because it suggests that these cells, and these SARS-CoV-2 specific T cells that are still present months out from infection, have the potential to homeostatically proliferate. And indeed, we actually directly demonstrated their ability to homeostatically proliferate using a CFSC assay. So essentially, we took the cells, labeled with CFSC, and then treated with IL-7 so that we could see which cells we could identify those cells that could homeostatically proliferate in response to IL-7. And then we looked amongst those cells to see which ones were SARS-CoV-2 specific, whether any were SARS-CoV-2 specific. Indeed, we could find some that were SARS-CoV-2 specific. And so this was kind of exciting because it suggested that these SARS-CoV-2 specific T cells are not only long-lived, but they can regenerate themselves. And it received a little bit of press attention because of the kind of good, kind of the encouraging news aspect of it. So this was fun for the lab because we appeared in the San Francisco Chronicle. This is two of my postdocs. We don't usually appear in newspapers, but essentially it's the take-home message is that these cells tend to be long-lived and they can homeostatically proliferate. Now, more recently, we've moved on to not just characterizing individuals that have recovered from mild infection, but also those that are hospitalized for COVID. And so in this follow-up study, which we recently put onto the preprint server, we are comparing mild, moderate versus severe COVID-19 cases. And again, looking specifically using Cytometry at the SARS-CoV-2 specific T cells. And so here I'm gating specifically on the SARS-CoV-2 specific T cells, and I'm comparing it from individuals with mild infection, moderate, meaning that they were hospitalized for COVID, but they weren't in the ICU, versus severe individuals. These are ones that were in the ICU, in the COVID ICU. And visually, what you'll notice is that these SARS-CoV-2 specific T cells appear to be in different regions of the TSNE, suggesting that they are phenotypically distinct. And I won't have time to go over exactly what's different about them, but one take-home message here is that the SARS-CoV-2 specific T cells from the severe patients tended to be more dysfunctional in the sense that they were more exhausted and more prone to apoptosis. But what we decided to focus our attention on in this particular study was to focus on the severe individuals, because amongst our severe individuals, about half of them survived after, severe COVID, and unfortunately half of them did not survive. But we had their blood specimens, so we could use this information to try to better understand what enables someone to recover from severe COVID. And so here I'm showing again the data in the form of TSNEs. Here I've combined the mild and moderates into one group and show that in gray. And here I've separated the severe individuals into the ones that did not survive severe COVID versus those that did. And so two things that you'll notice. One is that the cells here are in different regions of the TSNE, suggesting that the phenotypes of the cells are different in the survivors versus non-survivors. And the other thing you'll notice is that there are fewer cells here. And indeed, when we look at all the donors we had analyzed together within the, from the ICU specimens, that the non-survivors had a lower magnitude of SARS-CoV-2 specific T cell responses compared to the survivors. Now, what exactly is different about the phenotypes of these SARS-CoV-2 specific T cells in these two groups? Well, I showed two of them here that were interesting. One is that the non-survivors, their SARS-CoV-2 specific T cells tended to express lower levels of CD127 compared to the survivors. This is the same CD127 that I mentioned earlier. And this suggests that potentially in the survivors, you have more CD127. So these cells are able to homeostatically proliferate. You can have more of these cells, which might help these individuals recover from severe COVID. Now, interestingly, we see the opposite effect when looking at the percentage of SARS-CoV-2 specific Tregs, where it's actually higher in the non-survivors compared to the survivors. And we don't really know what this means, but potentially it could mean that in the non-survivors, these SARS-CoV-2 specific Tregs could potentially dampen or limit the development of a more robust SARS-CoV-2 specific effector T cell response, although that remains to be demonstrated. But finally, we looked not only amongst the SARS-CoV-2 specific T cells within our data sets, but we also obviously have a lot of bystander T cells. So those cells that are not necessarily specific for SARS-CoV-2, but are present in these various groups of individuals. And we did find something interesting when looking at the bystander T cells. And that is that when we compared the non-survivors versus the survivors from the ICUs, we found that in the survivors, they tended to, over time, have a diminishing proportion of these cells, these T cells that were dually positive for the activation marker CD69 and the chemokine receptor CXCR4, which we're all familiar with because we're HIV virologists. And so this is just an example of one donor where we're looking longitudinally over time at 4 time points. You can see it's high early on, and then it decreases over time. And this is true for both the CD4 as well as the CD8 compartment. And what I'm not showing here is in the non-survivor, you see the opposite trend where it's low to begin with, and then it increases over time. And this is actually all the longitudinal samples that we had together, where essentially it supports the notion that in the days leading up to death, you essentially have a massive increase in the proportion of these CD69 positive, CXCR4 positive cells. And so this was interesting because CXCR4, in addition to being one of the HIV co-receptors, it also directs cells to the inflamed and damaged lung. And so we decided to look to see whether there was any evidence that CXCR4 positive T cells were elevated in the lungs of individuals with more severe COVID. And so we mined some public data sets of generated from the lung of moderate cases of COVID versus severe critical cases. And so we extracted the CD4 and CD8 data and also the epithelial just as a control. And so that this is where those different cell types are. And then we looked at CXCR4 and CD69, the 2 markers that I had mentioned in the previous slide. And indeed, we do find that in severe and critical cases, these T cells, which we assume a lot of them are bystander T cells, that they have elevated levels of both of these factors. And this was work that was done by my collaborator, Jun Kai and Leah Vergossen's lab at Emory University. Now, it was quite exciting to us to see this kind of massive infiltration of CXCR4 positive cells because there are antagonists, as we know, against CXCR4. And actually tomorrow I'm talking to a pharma company who reached out to us because they saw our preprint with the idea of whether a CXCR4 antagonist might be useful in treating severe COVID. So we're interested to see where that goes. And so this is kind of a visual summary of the various observations that we made with regards to SARS-CoV-2 specific T cells, as well as bystander T cells and their association with the various severities of COVID. And if you're interested, I encourage you to check out our preprint paper. Okay, so to summarize this second part of the talk, I described how our initial results with convalescent COVID-19 individuals suggested that long-lived SARS-CoV-2 specific T cells capable of homeostatic proliferation can be found months out from infection. And then also I ended by talking about our more recent analyses of survivors versus non-survivors of severe COVID and how we are doing extensive comparison of both the SARS-CoV-2 specific T cells and bystander cells and found some interesting associations there. And with that, I'd like to acknowledge the folks that did the work. I'd like to acknowledge that the in vivo latency project, which I spent most of today on, was led by Jason Neidelman in my lab, but in collaboration with many others. I think I mentioned others along the way, but Jason was also the one that was the lead on the COVID project as well. Also want to thank all our other collaborators and many thanks to, in particular to the SCOPE and CHIRP participants for providing all of the valuable participant specimens. And with that, I will be glad to take any questions or maybe. Thank you for your attention.
Thank you so much, Nadia. That was amazing. Your data are so impressive and we all love your work. So I don't know if anybody has any question for Nadia.
Well, if I may, my name is actually also Nadia.
Hi, Nadia.
And hi, I'm a big fan of your work with HIV reservoirs on the COVID data. Is also awesome.
Thank you.
I actually had a question. So when I was reading the paper that you guys published on the reservoirs on how you identified the panel with 8 biomarkers that worked more or less consistently to enrich latent infected cells, how did you come up with those 8? I think I missed something in the paper, but presumably you have 40 and then you identify the differentially expressed between your predicted precursors and the rest. But then you have a lot of this panels that just look like flow data 1, marker versus the other. So I wasn't sure how you came up with those particular 2x2 comparisons that led to the selection of those 8. If you could comment on that.
Yeah, no, great question. So it was through a lot of manual. So first what we do is we just limited to surface markers, obviously, right? Because we had a number of intracellular markers, so we first just got rid of those. So we can't look at that. And then we also have to consider things such as, so how differentially expressed is it on these predicted latent cells versus total memory cells? And we can do that kind of in a systematic fashion by looking at mean signal intensity. And so we kind of went through that. Then the other thing we have to consider is we only have 8 to 10 channels, right? And even those 8 to channels, we can't just stick anything there because there's also in flow cytometry, you have the whole, the spectral overlap issues. And so it was a lot of just kind of back and forth. And you know, our first panel wasn't necessarily the best, but I would say, yeah, there was just a lot of manual assessing of all the markers.
I was just wondering as a follow-up.
Also, sorry, just to comment, and also share between different donors, right? Because there are a number of markers that we found that actually, you know, really enrich for reservoir cells in this one donor, but it's completely wouldn't in another donor. So then we would eliminate that one.
Yeah, I was just wondering in your follow-up RNA-seq experiments, when you enrich and now you can potentially do the same thing and look at a lot more parameters because you have data about all the expressed genes and maybe finesse it a little bit. But then you will have so many parameters that I'm not sure how you would select. Because I'm actually doing RNA-seq, single-cell RNA-seq myself, and I actually have a little bit different approach. I'm trying to look at latency directly without reactivation.
Yeah.
But yeah, the question about selecting the ones when you have, you know, thousands of parameters, I think that's challenging.
Oh, you mean, yeah, you have single-cell RNA-seq data set is not, it's no longer 40, right? It's about
right.
Yeah.
So maybe we could talk about it later by email or something.
We definitely should. Yeah, we just started generating some data sets of these enriched populations, and we're looking at those markers. I think it would be great to discuss.
Thank you.
Nadia, this is Stephen.
Sorry, who is this? Hello?
It was Steven, but I think he was frozen. Steven?
Yeah, it looks like.
I think you froze.
Steven, can you record us?
Okay.
Can you hear me now?
Yes.
All right, I'm on Sarah's computer. I'm on Sarah's computer, that's why I'm over here. Anyway, thank you, it was a great talk. I had one question about the endometrial cells. It was an interesting project, and I just wondered, were those from biopsies for a particular study, or were they like leftover remnant cells? I don't know, how did you get that tissue?
They're biopsy. So we have OB-GYN collaborators that we have a IRB in place. And so these are uninfected individuals, in case clear there. So some of them have PCOS because oftentimes they, so we have kind of two main sources. One is individuals that have PCOS or endometriosis because they're coming in. That's why they would get the biopsy, and they agree to give some of the cells for analyses. The other source we have, although it's been harder with COVID, there is oocyte donors. And so those are just young, healthy women that donate a biopsy because they're coming in to be an oocyte donor.
Okay, well, I think that's a really interesting line of research there. Thank you. And I know Sarah just wanted me to mention, because she put it in a comment, but she had to step out, Sarah Gianella. So she wanted to thank you and say it was great work. And if it's all right with you, Sarah, Antoine, and I are all working on a project using some FlowSum to analyze flow cytometry data. And I'm going to send you an email. We'd love to sort of pick your brain about some of the things we're seeing, because it seems like that's an area of expertise you really have, and we're hoping we can get something working here. So
yeah, happy to. And I probably bring in some folks from my team as well.
Awesome. That'd be great. I'll reach out.
Sounds good. All right, is that it?
Well, if there is no more questions, I would like to take over for Sarah, and I actually asked if we could invite her because I didn't know you personally, but she did. So thank you for coming and giving that awesome talk about all your research projects.
Thank you. Have a nice day.
Thank you.
You too. Thank you.
Thank you.